Swayam Prabha. Digital India. Educated India.
Jnanam Paramam Dheyam. Knowledge is supreme.
Welcome to MOOC course on applications of interactomics using genomics and proteomics technologies. There are advent of new technologies which are really trying to provide high throughput solution and much more reproducible and reliable results. One way of doing high throughput assays is to tag the proteins of interest with different type of tags or reagents like fluorescent tags or radioisotopes or chemiluminescent tags, and then trying to monitor the signals obtained from them. However, if you are adding a chemical moiety on your proteins of interest, it may provide some artifact results. It may alter the protein binding or the protein functions as well. So, there is a lot of emphasis about how to do the protein studies in the label free manner, and there is the advent of new technologies which are trying to achieve the protein protein interaction, protein other biomolecular interaction studies by using label free platforms. In this light, today we have invited a guest, Dr. Shiv, who is the head of business operations at Nanotemper Technologies, to talk to you about a latest technology MST, which is MicroScale Thermophoresis, for quantifying the binding events and studying the biomolecular interactions. Dr. Shiv will also provide you a demonstration of how to use MST technologies for biophysical assays. Towards the end, one of the colleagues, an application scientist, will explain the basic principles of TICO, which is used to check the protein quality and purity in a very short time. So, in this manner you will have a good understanding about how to first test your proteins of interest, quantify them to check their quantification and also their purity level, and then further study how they can bind with another protein of interest or a drug molecule of interest. So, let us have this lecture with Dr. Shiv.
I will quickly start my presentation without wasting much time, but before that I would like to know are you doing any biophysical assays for the interaction studies, protein, small molecule? If you are working on those lines, for sure this talk should be very interesting to you. So, I am sure you are working on those lines. Okay, great. Okay. So, this is something like a biophysical technique that determines the binding affinities. So, as I said this is a technique about a binding affinities, how you determine the binding affinity between two biomolecules. It can be anything, protein, small molecule, protein ligand, like anything you can determine the binding affinity. So, but before that I would like to quickly introduce myself as Shivramaya Nallapeta. You can call me Shiv, currently taking care of Nanotemper business in India. We are based in Bangalore, and my other two colleagues, Sajid Median, application specialist, and my other colleague Ram is also here with us. So, before going into the details of the technology, I would like to give a quick introduction on how our company has formed, which would be very interesting. The technology what I am talking about is thermophoresis. So, thermophoresis, have you ever heard of it before? Yeah, we say that whenever there is a heat, the molecules will move from hotter region to the colder region, but it will be a directed movement. So, this principle was discovered by these two gentlemen 150 years ago, but there was no any application. It was only a physical principle, but there was no any application until the two CEOs of our company who filed a patent in 2008. They said that the thermophoresis can be established at a microscale level, which can be used to determine the binding affinities. So from there the company has started, and as I said, like the first instrument which was launched in 2010, which is in front of you, they said that using this thermophoresis principle you can determine the binding affinities. Later on different versions came into the picture which I am really not interested, but other than this we have one more technology which measures the protein stability. Like you just wanted to know till at what temperature your protein is stable, like what you call the TM, melting temperature of your protein. So these are the two technologies what company has, and as I said, like these two instruments are very handy just beside me on that side and this side. Maybe during this talk is over, we'll also have a quick demonstration and we'll see how easy it is to run a experiment without any optimization. So these are the two CEOs of our company. As I said, like they are 31-year-old who has actually filed a patent and started this company in 2010. And now we are successfully spread across the globe. And as you can see a coffee machine in between them, we say that our machines are very simple. They are like a very robust, like a coffee machine. Like it's a vending machine. Like you want to do an assay, everything you just prepare and keep it, and 15 minutes of time everything is ready on your table. We'll see it as well. So with this quick introduction of the company, as I said, like it started in a small garage in 2008, and we slowly spread across the world, and we have more than 700 instruments across the world. And the best part of this company is like you see from 2010 to 2017 we got 1700 publications in all reputed journals. So that is how we had been connecting more to the scientists. It's completely a technology driven company, and it has been estimated that 15 million experiments are done using our technologies till date. So I think this is the more than introduction of the company what I can give. And now I am just coming to the technology. As I said, it is a thermophoresis. We have established at a micro scale, at a very small temperature gradient, we have demonstrated this thermophoresis. This is called the MicroScale Thermophoresis. In a short form you call it as an MST. So but before going into the details, I just want to elaborate more on the basic principle. What is thermophoresis? We all know what is electrophoresis. So in the temperature, in the electric field, the ions or molecules will move based on their sizes. If there is any gradient in between them, like an acrylamide or like any kind of a gel. So similarly, thermophoresis is the movement of the molecules in the temperature gradient. Thermo means temperature, phoresis movement. The movement of the molecules in the temperature gradient is called as a thermophoresis. But this thermophoresis is sensitive to for three things: size, charge, and the hydration shell. So as in electrophoresis you are only seeing the size, right? But here this movement is dependent on three important things: size, charge, and the hydration shell. So if any of these things changes during the molecular interaction, the thermophoresis behavior changes. So that is the basic principle what we have been using it. Maybe I'll go more into the detail. Size, charge, and the hydration shell. So normally when a protein binds, the size changes. When a protein DNA binds, the charge changes. And when a protein small molecule binds, the charge never changes or size never changes, but still one water molecule is displaced upon the binding. So it all depends how it is binding and all this stuff. So this technique has become very sensitive for all these three parameters, and there will be a change in the thermophoresis behavior when there is any change in these parameters. Yeah, so generally, as I said, like thermophoresis, the moment you put a heat, all the molecules move away from the heat. That is a general principle. As you can see here, this is just a cover slip slide which has been coated with a LMU. The moment you are shining with a laser, all the fluorescent molecules are moving away. So that's the reason why you are able to see the LMU. This is a Munich University. So that is called as a positive thermophoresis. The molecules moving away from the heat, which is a general property, is called as a positive thermophoresis. Similarly, there will be like the vice versa of it, what you call it as a negative thermophoresis. Maybe I'm not able. Sorry. So in the negative thermophoresis, what happens is the molecule move towards the heat. So it is like example glass beads. When you shine some laser or when you heat them, all the molecules move towards the heat. That is called the negative thermophoresis. So in principle, when you are doing a thermophoresis experiment, you always see these two kind of a phenomenon, like either they move towards the heat or they move away from the heat. But in our principle, you can measure both their movement. How we measure, maybe I can take you to the next slide. So if you look into the first slide, the two gentlemen who has proposed the theory of thermophoresis, they state that the steady state concentration of the molecules before and heating are directly dependent upon the three parameters, that is size, charge and the hydration shell. So as I said, before heating, whenever you are shining some heat, the movement of the molecules that has gone towards the hotter region and the molecules in the colder region, their ratio is always dependent upon these three parameters. That is what the law of thermophoresis says. And all our experiments have been highly validated and it follows this principle. So with this basic principle of thermophoresis, we built up an instrument which has two important things. One is a laser which creates the temperature for thermophoresis, and here one of the binding partner is fluorescently labeled. So just to see the fluorescence, you have a fluorescent detection unit. And the biggest advantage in this technique is you are not immobilizing anything as in other techniques. So you directly determine the binding affinity in the buffer, and you require only 4 to 5 microliters of the sample and the time. So normally you do a dilution series up to 12 to 16 dilutions, and every dilution take 40 seconds to scan. So in 12 to 15 minutes of time, you are able to determine the binding affinity without any compatibility with the buffers. There is no any crosstalk, there is no any buffer interferences in your assay. There is the biggest advantage of this technique, and many people ask the temperature you are creating, how much is the temperature we create? It's a very steep gradient, as you can see in the slide, 20 to 23 degrees. So that temperature doesn't affect any of your molecules. So in that way, what we do is you fix one of your partner and titrate the second one of a different dilutions and irradiate with the laser, and you see how the biomolecules move away upon the interaction and when they are not interacting. So that differences will help us to determine the binding affinity. So let's see more in detail in the next slide. See, if you look into the typical signal, what is actually happening? As I said, you are fixing one of the partner and the second partner you are titrating it, right? So on every concentration, on every dilution, whatever you are taking it, you are irradiating the laser. So as you can see here, for the first 5 seconds, all the molecules are uniformly distributed. This is generally what is, in principle, it is there. The moment you put the laser on, IR laser on, what is happening is all the molecules are moving away from the heat. And it states that within 30 seconds, they attain the steady state. So for 30 seconds, I am putting the laser on, okay? So what is happening is all the molecules are moving away from the heat, and within 30 seconds, they attain the steady state, as you can see here. And after that, I am putting the laser off. Since there is a fluorophore, again, there will be a regaining of the fluorescence. So again, your signal will shoot up, right? Fine. So now I have a sample where one of the binding partner and a different concentration of the ligand is mixed, and that has been taken in a small glass capillary, and on every glass capillary I am taking this thermophoresis reading. First 5 seconds it is very uniformly distributed. After 5 seconds you are putting the laser. Till 30 seconds we assume that it attains the steady state, and after that you are putting the laser off. So in this way, for all the different dilutions, whatever I am taking it, I will collect the thermophoresis signal, right? So this is how typically, as I said, in the top you can see, and each and every glass capillary, I have a partner along with the ligand, which is a different concentration. So now every of the glass capillary is going for the thermophoresis measurement. You see here. So there is a change in the movement. Can you tell why this change is happening? Yeah, upon binding, either the size or a charge is changing. So that's why the movement is also changing. If the movement is not changing, that means that there is no any binding at all. Then you have a signals all are overlapping one over the other as a clutter. But here, as you see here, as and how there is a binding, the different concentration of the ligand is coming, the movement is changing, right? So this is how we track the difference between the unbound state and the bound state. The unbound is where it is not binding, and in the bound state is where the biomolecule is actually interacting. So in this way, we are actually capturing the small differences in the thermophoretic behavior to determine the binding affinity. So once you determine all the 16 thermophoretic curves for all the different ligand concentrations, you just go to the dose fit curve. As you can see here, this is the baseline where typically there is no binding, and here this is saturation, and the midpoint is generally as a KD is what we call it as a binding affinity. So where your molecules are half bound and half unbound is called your KD. So typically by just extracting the data from the thermophoresis curve, we are fitting the dose fit curve to get a KD value. So this is the basic principle what it is underlined in the instrument. Just by the thermophoretic behavior changes from the bound state to the unbound state, you are able to determine the KD. Since this technique is very sensitive for size, charge and the hydration shell, you always get a differences in the thermophoretic behavior which will help you determine the binding affinity. The biggest advantage is you are taking a smaller volumes and you have no interferences with the buffer, and there is no any limitation with the sizes. So this is the basic principle. I am sure if you have any questions you can stop me here. The rest are how the examples how protein interacts. I have some good data I want to show to you. So this is the basic principle how this instrument works and how we determine the binding affinities. So with this you see a wide variety of samples, so from ionic interactions to the liposome interactions, nanodisk, any kind of interactions can be studied in this instrument. So there is no any limitation of size here. There is no any limitation of any of the properties of the molecule here. So anything, even like ionic interactions, you can imagine their ions are almost in a Daltons, and even if liposomes or ribosomal complexes, they are in a megadaltons. Even if there is an interaction between these things, still the techniques works very nicely. As I said, we got 1700 publications in the last 5 years. We have got all the supporting data with all different kind of interactions we have. So if you are working on a specific like protein small molecule, you can still discuss. I can help you out how we can do the assay and other stuff. So this is the main USP of this technique. Without any limitation of the sizes, you can work with a wide variety of the samples to get the binding affinity. So typically this technique is comparable to the SPR. Have you ever heard of SPR? I think many people might be using the SPR as well. So this technique is very much comparable to the SPR and ITC, isothermal calorimetry. So in the chemistry, people will be just measuring the heat exchange to determine the binding affinity. So the biggest advantage over all these techniques is, as I said, we did a protein DNA interactions, and you see in all the 3 techniques you are almost getting the same KD. We got 29 nanomolar, SPR 24, and ITC we got 30 nanomolar. So whatever the binding affinity you determine in the MST is very similar to the ITC and SPR assays. But what is the biggest advantage? So as we are all familiar with the SPR, you always have a chip, you immobilize one of your protein and pump your different buffers and ligands, and it involves a lot of method development and method optimization, and of course the cost for every assay. And in the ITC, we know you have a huge pump, and then you need to fill your pump and then start putting it up. So there higher volumes of the sample is required. So if you look into the biggest advantage of the MST is you don't require the higher volume of the sample. We look with the always the lower concentration of the sample, and the assay is done in 10 minutes of time with no cost. The glass capillaries are only the consumables what we use for doing the assay, and it will cost you €5 per assay. So at the end of the assay, if you look on to perform 1 KD measurement, you require €5. So that is the biggest advantage, the smaller volumes, time, and the overall cost of the experiment. So this has been highly worked out with all the 3 experiments. As you can see here, the cost it's almost like 1/10 is what we generally do it for an SPR and other assay experiments. So maybe people working with SPR still might have some questions. You can still feel free to ask me. So as I said, we do a labeling to one of the binding partner. So we recently came up with a kit where you can label your his tag. So at the 6 histidine position, you can label your protein. So in such a way, what is the biggest advantage is you need not purify your protein. If your protein is in the cell lysate and it is already his tagged, just simply add the dye and do the titrations. So in that way, even in the cell lysates, if you want to have a specific interaction studies, this can be successfully done. So now I'll just take you to some examples of how this application has been used in a different variety of interactions. One is the dimerization. So when you are talking about the protein-protein interaction, the same protein, when it interacts, it becomes a dimer. So it's extremely difficult in other techniques like maybe in SPR or in ITC. When the same protein you are immobilizing and same protein you are running through, you get a lot of artifacts. So but here, labeling one of the protein and titrating the same protein, you can determine the dimerization KD as well. As you can see here, you can clearly see a clean fit of KD coming up here. So even the protein dimerizations and protein dimers can be easily studied using this technology. And other most interactions people are interested in is protein nucleic acids. So I'm sure you are well aware of this CRISPR mechanism. So here the RNA to the nucleus protein has been studied, and this paper hit the Nature, and here in this paper they quoted that this is the only biophysical technique that has been to determine the binding affinities. So even the cutting edge technologies also MST has become a much handy tool to determine the binding affinity. So maybe traditionally people does EMSA. So have anybody done EMSA here? Oh, great. So you know what's the pain of doing an EMSA there, and you always get a qualitative, you never get a quantitative information there. So in the same studies which are done with the EMSA, we try to label the DNA like a Psi5 or Psi6 or whatever you have the dye, and then titrate your other things, and you are able to determine the KD which is very much similar. So you are, with a smaller volume of the sample, you are quantitatively determining the KD over this, all this EMSA and radioactive labeling, everything is all been overlooked when you do this kind of assays here. So other example is a protein ionic interactions. These are always a very special interactions. As I said, when a protein and ion interacts, there is no size at all. There is no any size making any difference. The ion is a very small in size and your protein is a very big. So that time the size doesn't change at all. But still when a protein goes and binds, there is a displacement of one water molecule at least outside the protein or anything. So even with this small change, we are able to determine the binding affinity. This is the biggest advantage. As you can see, this is one calcium binding protein which has been done, and when the same experiment was done in the presence of a magnesium, there was no binding. As a positive and negative control, this experiment has been done. And as you can see here, this is from the Max Planck Institute, the person who has developed the ITC, who is known to be the father of ITC. So always when you are working with the nanomolar, picomolar interactions and a very small size molecules, this addresses very nicely over any other techniques. So and again, this is a protein small molecule interaction. Here you have a ribosomal protein complex in the red color, which is few megadaltons, and a small molecule which is about 120 daltons, the cancer drug. So when it binds, the size is absolutely not making any difference. If you can imagine megadalton protein and 120 dalton small molecule interacts, the size doesn't change at all. But in principle, if you look into this structural information, the water molecule is actually being displaced, which we call it as the hydration shell, because of which we are determining the thermophoresis change and the binding affinity. So, and other experiments, many people who are working with the enzymes, they might be more interested with the this kind of what you call the inhibitor studies, inhibitor screenings. So generally, as you know, you do a binding with one specific what do you call this, the tracer molecule, and once you know the KD of it, and again you titrate the second one. So wherever it displaces, then you always know the binding. Competitive assay experiments can also be easily studied. We have a well robust software to support all this kind of studies at the end of the day. So this is again the same example with high molecular weight protein complexes. As I said, few megadalton proteins, when interact with the proteins, still you are able to determine the binding affinities very easily. And the other challenging thing is if anybody is working on the membrane proteins, hope you know what is the pain of making the membrane proteins more stable. It requires a detergents. So any other techniques when you have a detergents, you always have a buffer incompatibility. For suppose if I have an STS, if I have some a foaming agents in your things, then obviously you get a lot of artifacts. So here in this technique, as I said, we are using only 3 to 4 microliters of the sample, that thing can be nullified. So even membrane proteins can successfully be studied here. As you can see, the paper again hit the Nature, where the membrane bound protein has been expressed with the GFP, and that particular protein has been studied for the microscale thermophoresis. So in this way, any variety of sample, it need not be a protein, any kind of molecular interactions, protein carbohydrate, protein ion, anything in between them can be studied using the microscale thermophoresis. As I said, this is one example where the cell lysate, like when you are working with the biological fluids like maybe a serum, cell lysates, you have thousands of protein. So you can even bypass by doing them not doing any purification. If your protein of interest is always with a his tag, you add the dye which is very specifically binds to the his tag and then titrate the other partner. So in this way, even for disease markers, many people working with the proteomics are also using the MST as a complementary tool to screen the different samples for the analysis here. So this is again just the example like how we get the data here. So as I said, like any one of the binding partners should be labeled. It can be A or it can be B. There is no mandate that you should label only a protein. You can label anything. You can label a small molecule, you can label a protein, any partner can be labeled, but the binding affinity doesn't change at all. So that is the main USP of this. So this has some examples what I want to tell for a variety of interactions, how this techniques work so successfully. So any questions or any other things you wanted me to know, I shall be glad to address. So now just as I said, you are aware of the principle, how it does it, and you know what variety of applications can be done on this. Now I'll just quickly take to the software, like how this instrument, like how it can be operated and all these things. Maybe in a nutshell, I just want to tell this software is an artificial intelligence driven. So that means that the moment you set up the assay, it runs on its own, and once the analysis is over, the software automatically assesses whether this data is good enough or not. Is there something like still you are not getting a proper results, or because of any reasons, it will automatically give you a suggestions. Okay, if this is not a binding fit, then it will tell you please change the buffer, you change the pH, or you change your the protein concentrations. So like this automatically software does the assessment. So in a way it is very much easier. Of course, as a user we always know technically what is happening with the software. By looking to the data we can always assess, but still software will help you to make a judgment on your analysis, and you always get all the data in a single page. As you see here, this is a clean binding fit. So and these are all the thermophoretic traces of all your dilutions what you have made it. So first suppose your samples are making an aggregates. So for suppose upon a particular concentration, when it is interacting with the ligand, it becomes an aggregates. So then aggregates means then the sample will be non-homogeneous. The thermophoresis behavior also will be very random. The curve looks very random. I'll just show how it looks like. See, whenever there is an aggregation, see, you see here that the curve is completely a wavy. So like this qualitatively we can always judge the aggregation of your sample also. So in other experiments, when a sample is aggregating, it is already in contact with the instrument, it spoils the next run, it spoils the next things. So the biggest advantage here is you are using a different independent capillaries for every dilution. So if one capillary is having an aggregate, you can just discard them, and you know at what concentration your protein is actually aggregating. So in this way qualitatively you can judge the aggregation in your samples by using this kind of a technique. So maybe all these things I don't want to bore you much, because as I said, it's more for users who are actually doing it. In principle, the software is completely a artificial intelligence driven. It gives you suggestions at the end of the day, like how you need to move on. So this is I just want to summarize quickly. We are already associated with the top premium institutes across the globe, and I am very glad that in India we are associated with the IIT Hyderabad, RCB, Novozymes, and many other customers who are using this technology. And as I said, in all kind of journals you can find the MST papers. Like in Nature we have more than 15 papers, Science, all the best journals have acknowledged this as a biophysical tool that actually be a very useful tool in measuring the binding affinities. So here are some customers who had been using a lot of SPR, ITC experiments and now made a shift to the techniques and found how easy it is. Like if you are doing any screening, you are doing a drug screening, you have a small molecules libraries, hundreds or thousands of molecules. So I'm sure like other techniques will be a nightmare. So here, since you are using it in 10 to 12 minutes of time, you can always screen them very quickly. And these are our some customers who has used it and gave a very good feedback. And we have more than 10 publications from Indian customers in the last 24 months. The final slide is always what I say. As I said in the beginning, it is a company of scientists. So along with it's not just telling about the instrument, it is all about giving a good support. So you're always bundled with a good scientist to support you and develop the assays on your own. So that's all I want to tell. Since we already are going to give a quick demo, maybe like, we can just quickly share some questions you have, or if you are working on any other things, what are the challenges you are facing, I shall be very glad to help you out. What kind of samples you do on SPR basically at the moment? ITC, okay. So for sure, like, as I said, like now we have a positive control kit. It is a DNA aptamer and a small molecule, ATP, basically ADP and the DNA aptamer. So anybody is interested, they can just come in front and then just do it, because as I said, like this doesn't require any much training, much thing to understand. As I said, the basic principle is thermophoresis, and how every dilution there's a difference is what we need to understand, and rest everything is done by the software automatically.
This protein and small molecule interaction, like protein and this alcohol.
See, that's what I'm saying. In principle, anything is possible. Alkaloid, can you tell me an example? Curcumin. So very recently, I think, as I said, like 15 minutes of the time is the measurement time. So maybe people can come front and just wander, or you can sit there and see however you wish. So we have a control where we are just going to load into the capillaries and do the like an MST assay. And we'll go to this one, the stability one. So there also the things are ready. It takes only 3 minutes to measure the stability of your protein. That is the biggest advantage. So maybe she will give a small talk on few slides. What are the basic principle and what are the applications? So anybody working with the protein can use this instrument for quickly checking, like, the functionality of your protein, whether your protein is really functional or not. Okay. So these are the glass capillaries, what we use for the assay. You can see these are the simple glass capillaries, which can accommodate maximum of 5 to 6 microliters. You just dip into the solution. So normally you take a PCR strips and then, like, fix your partner A and titrate the ligand. Like generally like 12 to 16 dilutions is what we prepare, and all the 12 to 16 dilutions, each dilution you take in the glass capillary. So and this glass capillary will go for the measurement. So and here you have already, like, we already mixed the DNA aptamer and the ATP here. So what I'm just going to do is now, I'm just dipping the glass capillary and then loading it. So it's all like once you load it, within 10 minutes of time you can do it. So normally, proteins are very like, unstable at a room temperature. So the DNA has been used. The DNA is very stable here. So generally for demos, I prefer to do with the DNA because it's more stable. You can carry anywhere what you wanted. Yeah. So this is a tray which holds your glass capillaries. So it's like labeled 1 to 16. So like whatever you are taking it, you just try to put into this and then do it. And we have a small magnetic strip to arrest your glass capillaries. It's like, when you are loading it, they might not fall down. So it's just a magnetic strip to arrest the glass capillaries when you are loading it. That's all. So maybe we'll take 2 more minutes to load, and after this we can just start the assay. Yeah. Yeah, if somebody want to load, they can come and load. You can load it, no problem. Or a protein purification by columns or anything like that. So how you maintain the quality, how you check the quality, that is nothing but the stability or the functionality. So we have recently launched this instrument, only not even 1 month old is this. So we can quickly check the protein quality. What is the basic principle of this and what kind of applications. She'll try to summarize and give it in just 15 minutes of time. And after that instrument also we can give a demo. It takes in principle only 3 minutes to run 6 samples. We got some controls, we can quickly run that also. Saji, please go ahead.
So have you ever asked to yourself that whatever protein you are purifying, whether it's pure or not, I mean whether that can be used for your future assay experiments or not? So that's a very big question that always comes in the mind of any researcher. So before going for any assay, what you do is like you run a column, you do a SDS page, or you do spectrophotometric analysis. So, these are like very standard gold methods, like going for a spectrophotometry or electrophoresis or a column chromatography, but still you are not aware whether your protein is a functional protein or not. So, we have this newly launched product which is known as Tycho, and in one go it takes 6 samples and you can actually check the quality of the protein. So, it will come to know whether the protein what you have purified is functional or not. So, what is the basic principle of this machine is that it can work with any kind of protein, whether it is an antibody or whether it's a normal protein, membrane protein, receptor protein, any kind of protein, and it give a 3-minute measurement. So, within 3 minutes you are actually checking your quality and the purity of the protein. So, it is a label free measurement, like here one of the partner is labeled, you are not labeling here. It is the intrinsic protein fluorescence that we are checking, which will give you a label free what you call inflection temperature, which is very equivalent to the melting temperature. And you are similarly using capillaries, and the amount of the volume that you are using is less than 10 microliter, and in one go almost 6 samples you can analyze. Then the run rate here is little fast because you are analyzing in 3 minutes. So, the run rate here is 30 degree per minute, and as of our all the instruments are maintenance free. There is no cleaning, you are just putting the capillaries in the instrument, taking off once the run is complete. So, what are the basic benefits here is that it is a faster measurement technique, and it checks the purification and the checks at the purification and the characterization levels. So, if you have like batch to batch samples or like 1 day old samples or 10 day old samples or like very old samples, and you are freeze thawing the samples, all that samples when you take and check one by one, you can check the actual purity, whether it has denatured or not. Like that you can always check batch wise. Then you can analyze the samples in a wide range, that is from 5 microgram per ml to 250 mg per ml. And as you can see, it is a very small instrument, you can hand carry the instrument here and there. So, it is a very easy to work with. So, when you see the characterization workflow, where all the Tycho can be used. So, when you go for the initial purification, after that when you get the protein, you can check the purity at this level. After chromatography, ion exchange chromatography or column purification, you can again check the purity of your protein. Once the protein is purified, then you have to store it at different conditions. So, at which buffer, at which pH you want to store it, you can always check that and then store it in that conditions. Once you have stored the proteins, suppose you have kept it for 10 days or 20 days, after that you want to again check the purity, because for assay you need to have a pure protein. If it is not pure, then definitely suppose you are going for SPR, one chip you can't waste if your protein is not pure. So, before the assay development, you always have to check the protein, how the protein is. If it is totally denatured and you are doing an assay, definitely you are losing your time as well as the chip what you are using for a SPR. So, all these are the steps where you can actually check your protein quality. Now, I have few applications where it will tell you where all Tycho can be used. Now, in this, when you see what is the basic principle that is applied here, is that it is checking the intrinsic tryptophan fluorescence. So, protein from a folded to an unfolded state, it will give you a particular Ti value. So, the temperature, the results that a Tycho is going to give is the initial ratio. The initial ratio is the ratio where it will give you the purity of the protein, whether it is denatured or not. And the delta ratio is the ratio which is from the folded to the unfolded region. Like that, when the protein folds, when the protein unfolds, you will get a TI value as well as the sample brightness. The sample brightness here will give you a relative concentration of your protein molecule. It will not give you actual concentration, but a relative concentration that yeah, this much amount of protein is present. And then protein profile similarity is that if you have a reference sample of your protein, that can be taken as a standard protein sample, and then when you are going for batch to batch, or like if you are doing a freeze thawing, at that time you can always take that as a reference sample and cross check the other batch to batch, or you can freeze thawed ones and then check how much it has denatured or how much it has changed. Now here is one example where you have compared it with batch to batch screenings. So here you can see the protein which is a pure protein and a protein that has been kept in -80, it is almost showing the similar profile. But when it is used after 1 week, but kept at 4 degrees centigrade, the profile has changed. So you can see the shift has gone up, that means the protein has slightly unfolded. So that means you can definitely go for assay, but still you can check the quality of the protein, how it has varied. Now here you can check the buffer screening as well as the pH stability measurements. So you know like basically for SPR you need low pH. So if you have a standardized form of how to do it, you can check at pH 4 it's it's totally denatured, and then at pH 5, which is like a basic pH for SPR measurements, the curve is almost good. And when you compare it with different buffers, you can always check which pH you can use, which buffer you can use. Now here you can also check the thermal shift assay. I mean, a protein is there, and if you want to check whether the protein can bind with a particular ligand or not. So when you run the protein without the ligand, you will see a particular profile, and then in the presence of a ligand, the shift will change. So the shift can either be a positive shift or a negative shift. If it is a positive shift, that means it's a true binder. If it's a negative shift, that means it's not a true binder. We can actually go for a yes or no that whether the shift is happening or not. I mean whether the interaction is possible or not. With this you can go for further assays, like if you are going for SPR or if you are going for ITC, you can say that, oh okay, this protein with this ligand it's good because there is the thermal shift, because the TIs are shifting here. Now, I have few examples which will show you the data. Now this is a example in which the monoclonal antibodies was checked at different oxidation levels. So you can see at the native state it has given a different profile, and then a 3-hour oxidation with hydrogen peroxide, it has given a different profile, and a 18-hour oxidation, it's giving a different profile. So you can see there is a TI shift in all the 3 samples. And when you had gone for the MST assay, the same thing you can see. The shift is, I mean the KD value is changing with respect to oxidation. Now, if you want to go for a biosensor assay like the SPR, so here you have to check the pH values. Definitely which pH is best for the SPR. So you can always screen different pH ranges and check which buffer is best. So like here kinase is taken as a standard, and at different pH it's tested. So you can see at pH 7 and pH 4 and pH 4.5, it's just giving a planar line. That means it's already denatured. Okay, but the other in pH 7 and pH 5.5, it's giving a curve which is showing the stability of the protein. Now this is a thermal shift assay that we have done, where we have taken a kinase, and then the kinase is treated with a ligand molecule. You can see the shift in the assay. You can see protein alone was giving a shift at 50-55, and once it's added with the ligand, the shift has gone to 62. So, that is how you can go check the thermal assays. Now, this is once the chromatography is run. I mean, you get lot of fragments. So, you check all the fragments and see which is your best, which is the best fragment where your protein is there. So, this will give you a protein profile that this fragment or the first fragment or the second fragment is having the best protein. But if you have a standard reference along with it, you can always validate that the protein is actually the pure protein. And now, here is another example that we have conducted, where you have taken fresh samples, freeze-thaw cycle samples, and fully denatured and partially denatured samples. So, you can see how the trend has changed when you are checking the unfolding profiles. So, the fully denatured sample is giving you a very planar profile, whereas the partially denatured is giving a different profile compared to the freeze-thaw and the fresh samples. So, the same is analyzed with the MST. So, you can see the fully denatured sample is, there is no KD value to it, but the other samples you can get a KD value, but the KD value will vary compared to the fresh sample. So, in all, if you see, it is a very quick tool to determine the purity as well as the stability and functionality of the protein. And when you want to characterize some kinases, some receptors, or some membrane proteins or transmembrane proteins like that, you can always check the purity of those proteins. And like it will give you a yes or no signal when you want to do a thermal stability assays, or you want to go for SPR assay or a cryo-EM assay or NMR studies. So, before that you want to check whether your protein is stable or not, whether your protein is pure or not. So, a yes or no will always, this machine will always help you with the yes or no decisions. So, I can give you a small demo with one sample, that is the BSA. That is 1 mg per ml I have taken. So, I can just give you, it is a 3-minute run that will be there, and we can check the profile how it goes.
Question. Do you have any example of this long-term reference proteins, whether they change in these assays?
Yes.
Yeah, so you have tested many sample proteins, the reference proteins that are used. For example, if you said NMR.
As such reference proteins we do not have, but in, yeah. But like if we do for you, yes. So, like if we have a reference protein, we can always, if you give us a fresh sample, if we have a standard protein, we can always correlate with that.
We say a positive shift is towards the right side and a negative shift is like towards the left side. The left side is always destabilizing your complex or your protein.
Yeah, but it's not supposed to shift.
Yeah, in principle, no.
In principle, no, yes.
Storage conditions or so many, what do you call this?
Because in few of the demos we have seen this kind of shift, where there is a positive shift as well as a negative shift. So, positive shift, yeah. So, the positive shift will always say that it's a binder, but the negative shift is destabilizing your protein, and you can see the profile when it goes above 0.9 or something, whether it is fully denatured or not.
When we store a protein, it will be denatured as days pass on. It will somewhat, it will be unfolded. It will not be in a stable condition throughout the time. Basically, the protein structure is like that only. It is not at all
That's
stable.
what your statement is very, I cannot say yes, I cannot say no. Actually, it all depends upon your specific protein, what you are working.
Yeah, again, when we do the assay process, you will not get same result. Somewhat it will be down only the result compared to the original.
Exactly, that's the main purpose.
As long as like 10-20% is fine. If your protein is unfolded more than 30-40%, then obviously you are working with the dead protein, which doesn't make any sense. Like for suppose you are immobilizing your protein and you are changing lot many buffers and you are trying to tweak everything, and if it is not working, the problem is your protein is really not in a a good state actually. A quick checkpoint can always be taken care of, and when you are doing a purification, like in all the fractions, you need not go to the STS page and then check every time. So quickly you can always check it, and you are storing it and you are going for the next assessment, you can see it. So it's basically like a quick checkpoint to know what is the quality of the protein what you are working with. I'm sure like till now you don't have any direct method to measure this kind of quality. You do the quantitation by spectrophotometry, Bradford, other things, and you do a color purification, you run the STS page, but there is no any checkpoint. You have to know that the protein you are working is really stable
Pure or
it's
not.
functional or not. So when you work with the bioassays, unless your protein is not functional, there is no point in setting up the assay and spending a lot of time. And since it's a label free, you don't require anything you just dip it and then do the measurement. So that would be the quickest way to check the protein quality.
And many times when you take the proteins out, I mean you keep it in ice, but still taking in out, so you know how whether the protein is stable or not. So this is like a quick tool.
She also shown that percentage unfolding, as you said, after 10 days, after 1 month, what is the percentage of unfolding? See, as long as 20-30% is fine. When it goes above 50%, then show your KD, everything changes. Then that is not your true protein, what you are actually working on. So on the background, we never know what is actually happening. When you are taking from -20 or -80 and thawing it, your protein might be spoiled, but you are not sure, but still you are going for the bioassay. If it doesn't work, then you think that there is something else wrong, but you don't know that the protein itself has been unfolded while doing the thawing or while doing storage.
And one more doubt, sir. Here it is, you are placing at 30 degrees centigrade for 1 minute. So at that temperature, protein will not get any.
See, our basic principle here is, when you see a protein, the tryptophans are actually buried inside, right? All your tryptophans are buried inside the protein. As now you are heating it up, the tryptophans gets exposed. Once your tryptophans are exposed, the fluorescence is changing. So when completely all the tryptophans are exposed, the protein has lost its the activity or the complete unfolding is happening. So that is the basic principle what we are taking in, and we are ramping 30 degrees per minute. From 30 to 90, we are reaching in 3 minutes quickly. Because just you are heating the sample, and once all the tryptophans are exposed in your sample, then you are getting a what you call the transition, TI, inflection point. So that is the basic principle. But we have other version of the instrument where you can get it 2 TMs, the melting temperature, 1 degree per minute or 2 degree per minute. You can always control the ramping heat and can get the TM values also. It's very similar to DSC. But this one is something for any bioassay, any mass spectrometer, you are working with any intact proteins. When you are injecting the LCMS, if the protein is already dead, then for sure you don't get anything. You just go and clog your nano LC needle or something like that. So here you can quickly check, hey, my protein is fine, so now let's go into the LCMS. So like this, as a quick checkpoint at a different experiments like NMR, cryo-EM, any kind of things you can always use this as a quick tool to know the protein quality. That's the main purpose of this. And cost effective and also 3 minutes, it hardly matters. 6 samples, you are getting the data in 3 minutes. Just you are heating it and then pulling it back. That's the main point. Is anybody working on DSC or CD or this kind of techniques? It's more mass spectrometry. Fine, so that's all. Quickly, maybe 3 minutes as I said, she will load 1 sample quickly. By the time she comes here, the time will be done there. So you just load 1 sample. Just we are showing the previous data to just get an understanding because we had only 1 BSA. So you can clearly see there is a change in the TMs, TI points. Whenever there is an interaction, generally it will always stabilize the complex. So there will be a shift in the TMs. So typically you always get the curves with a clear TMs, and if you just look at the results, you can always see, you always get the temperatures at which they got unfolded, and it's clearly showing a shift of somewhere 3 degrees, somewhere 4 degrees. So like this, you can always see the differences in the TMs. So that is the biggest advantage. Another thing is you are seeing the stability of your protein. For always, I know many times you purify the same protein over some months or something like that. So you can always compare what is the profile similarity, what is the percentage loss I'm doing here. So this kind of all things you can always integrate in your workflow to measure the protein stability. So you always get the delta ratio, which will tell you the percentage unfolding, the sample brightness. It is not exactly a quantitative parameter. It is kind of a relatively, if I'm taking 1 sample as a standard, against that one, what is the brightness, what you have it. It will always give the relative quantitation figure also. So it's very easy, like as I said, like at a point where you can always assess the protein quality for before going to any bioassay. It can be SPR, it can be like any ITC, anything like that. So you can always quickly know the quality of your protein, whether it is really good or not. And if it is good, then comparatively with your references, how it actually works on. That's the biggest advantage what you have to get. Because in one sample, it not make sense to show you all the data. So we have shown you the previous data. So this is how typically you are getting it here. The software automatically calculates and then show you. This is the TM of it like this.
If a sample is having 2 to 3 proteins in a same sample, can we place a sample of
So then it's, that it all depends, like when it is actually unfolding. So in some the tryptophans might be exposed, some tryptophans are like already buried inside. So in principle you get always a very non-specific data. Normally it's a biophysical, always work with a few proteins is recommended. But yes, many of our customers, like with 2 proteins they have tried it. So always they know, like one protein TM they know, the other one whatever it was getting for the other protein. It's always like R&D kind of thing. Sorry?
We can see that.
Yeah, absolutely, absolutely. And you can always see the multiple TMs in the same protein. It can have that, the same protein have multiple domains which are like, exposed at different temperatures. Like particularly maps, if you see, you get the Fab region and the FC region at different times. So like this, even if your same protein is having multiple TMs, you can still get the data. So this is the biggest demo what we can do. So this is how you get the typical results and all this stuff. So any questions or something? I'm sure, like here proteomics means everybody will be doing some kind of purification, mass spectrometry, all this kind of thing. So we shall be very glad. If you are interested, we can evaluate and we can still give more insights into the technology.
The advent of new technologies, both label based and label free, have now started offering us new insights for lot of new functions for the proteins of interest for which there was no function defined earlier. In this light, there is advent of new technologies which aims to do these experiments without tagging the protein of interest and also provide the binding and the kinetics information. In this light, there are new developments, new technologies are coming forward, and today we try to provide you interaction with one of the leading companies and their application scientists who talk to you about MST technologies and also the quality control assays were done using Tycho technology. I hope these provide you some insight about how to do your experiments for the proteins to further characterize using latest label free biosensors. Thank you.